Hello and welcome to another video lecture. In previous lectures, as you know, we discussed some basic concepts of cell signaling. Today in this lecture, we are going to discuss about G protein coupled receptors. First, we will try to understand the structure of these GPCRs, and then we will try to understand how they are activated and deactivated. So far as the structure of these GPCRs are concerned, they are present on the plasma membrane, not within the cytoplasm. Present on the plasma membrane, we have this transmembrane protein, and associated with this transmembrane protein, we have another protein complex that is called as this G protein complex. This G protein complex acts as a transducer. It is hired or it is recruited by this transmembrane protein to pass on the signal from this receptor to the effector molecules present within the cytoplasm. So we have this G protein complex and we have this transmembrane protein. Together these two protein complexes, they form this G protein coupled receptors. This transmembrane protein is a single polypeptide chain, and this single polypeptide chain, like any other polypeptide chain, is having two ends. One end is called as this amino terminal, having this amine group free, and another terminal is this carboxyl terminal having this carboxyl group free. This amino terminal is towards the extracellular side of the cell, and this carboxyl terminal is towards the cytoplasmic side of the cell. This orientation is specified at the time of translation, which occurs at the membrane of the endoplasmic reticulum when this ribosome is getting translocated to the translocon present the endoplasmic reticulum. So this orientation is fixed at the time of translation occurring at the endoplasmic reticulum membrane. So here we have this amino terminal towards the extracellular side, and we have this carboxyl terminal towards the cytoplasmic side. In between these two terminals, this N terminal and the C terminal, we have 7 alpha helices inserted in the plasma membrane. These 7 helices are 1, 2, 3, 4, 5, 6 and 7. And these alpha helices are having hydrophobic amino acids. So these hydrophobic amino acids having these alpha helices, they are inserted within this plasma membrane. And in addition of these helices, we are having these loops in the cytoplasmic side as well as towards the extracellular side. Remember one thing, that right from this N terminal towards the C terminal, this is a single polypeptide chain. You can say this single polypeptide chain has inserted 7 times into this plasma membrane, and we have these loops present in the cytoplasmic side as well as towards the extracellular side. These loops are connecting these alpha helices. For example, we are having this loop, C1 loop, present in the cytoplasmic side. This loop is connecting the helices 1 and 2. This helices 1 and 2, this is connected by the loop present within the cytoplasm, not within the extracellular side. Similarly, we are having this loop C2 present within the cytoplasm. It connects the helices 3 and 4. This loop C3, it connects the helices this 5 and 6. And we are having these 3 loops E2, E3 and 4 on the extracellular side. And this E2 loop, it connects this helices 2 and 3. And this E3 loop, it connects the helices 4 and 5, and similarly we are having this E4 loop, it connects the helices 6 and 7. You can say we are having 4 segments including 3 loops on the extracellular side, and we are having 4 segments including these 3 loops within the cytoplasmic side. This is the transmembrane protein, and as a transmembrane protein, it is having these segments which are in connection with the extracellular side of the cell, and these segments are in connection with the cytoplasmic side of the cell. As a transmembrane protein, it can easily receive the signal from the extracellular side through these domains, and it can pass the signal into the cytoplasm through these domains which are present in the cytoplasmic side. But here this transmembrane protein cannot change its location because it is permanently attached with the plasma membrane. How it can transfer the signal deep into the cytoplasm? What it does, it recruits another set of proteins for this function. That is, it sends it a signal through another protein. And that protein here in this case is this G protein. G protein acts as a transducer. It receives the signal through these cytoplasmic domains of this transmembrane receptor, and then it transfers this signal to the respective effectors. This G protein is called G protein because it binds with the GDP or GTP. Because it binds with the GDP or GTP, that is why it is called as the G protein. And this G protein is composed of 3 polypeptide subunits. One is called as the alpha subunit, then we are having this beta subunit and gamma subunit. This heterotrimer unit is attached with the inner leaflet of the plasma membrane through 2 lipid chains. Here we have this lipid chain. This lipid chain binds the alpha subunit to the inner leaflet of the plasma membrane. Similarly, we have another lipid chain that binds the gamma subunit to the inner leaflet of the plasma membrane. You can say this G protein binds to the inner leaflet of the plasma membrane through these 2 lipid chains. One is connected to the alpha subunit and another is connected to the gamma subunit. This alpha subunit can dissociate from this complex when it is bind with the GTP. When it is bind with the GDP, then it gets associated with this complex, with these 2 protein polypeptide subunits. This gamma subunit and beta subunit, they always remain together. They never separate from each other. This alpha subunit can dissociate, can separate from this whole complex, depending upon whether it is bind with this GTP or GDP. Here in this inactive state, it is bind with this GDP. That is why this is inactive state of G protein. When it is bind with this GDP, it is inactive. So this GDP or GTP binds with the alpha subunit of this heterotrimeric or G protein. It does not bind with the beta subunit or gamma subunit. Second thing you have to remember is that this heterotrimeric protein complex binds with this inner leaflet of the plasma membrane through 2 lipid chains, and these 2 lipid chains are bind with the alpha subunit and the gamma subunit, not with the beta subunit. So this is the structure of this G protein, and this is the structure of this transmembrane protein. This whole complex forms the G coupled protein receptor.